Hello, it's David Green, CEO of Regen Suppliers. Today we're discussing exactly what are exosomes. So first let's talk about what happens as we age. So as we age, we have a lot of body functions that start to break down. Tissue is getting older, it needs replenishing. So the demand continues to go up, but the amount of stem cells that we have to actually help perform those functions is diminishing. And not only is it diminishing, it's also a matter of those stem cells that we do have that start to get lazy. So the end result is we have this huge need and this really minimal supply, and that's not the best equation for success. So this shows that in a table form where you have, as a newborn, 1 in 10,000 stem cells in your bone marrow is a stem cell. By the age of 30, that drops down 25 times to 1 in 250,000. By the age of 60, it drops down to 1 in 2 million cells in your bone marrow as a stem cell. So it's almost like a needle in a haystack. And incidentally, this is one reason why none of our centers offers bone marrow stem cell therapy, because the numbers don't make any sense as we age. So when you look at what exactly an exosome is, it's actually not a cell in and of itself. It comes from cells, it's a byproduct of cells, and it could be any number of cells. You could see these cells in saliva, breast milk, semen, all types of body tissues and substances, but we're interested in them as a byproduct of stem cells, okay? So let's consider this being the membrane of a stem cell, all right? And inside, you have these vesicles that form called endosomal vesicles. So they're on the inside, endosomal, and inside those are what's going to end up becoming an exosome, as you can see here. So what happens is these endosomes eventually undergo what's called exocytosis, which is being discharged, and what's released are exosomes. Okay, so we'll get more into that in just a moment. But why are exosomes so popular? Well, the increase in interest in exosomes really got going around 2005 after this experiment out of Stanford, where they took mice who were either young mice or old mice, and they spliced together their circulation. Now, why did they do this? They wanted to see if there would be an effect of the young mouse's blood circulating over into the old mouse when they administered a muscle injury. And not only did they put young mice and old mice together, they put young to young and old to old, as controls. And what they found is that the young to young, they healed up the muscle injury tremendously. The old to old did not heal very well at all. So those were exactly what was expected. What they found, when you take on the left would be the young mouse, on the right would be the old mouse, when they looked at this and they administered a skeletal muscle injury to both, they found that the young mouse continued to heal those tremendously. The old mouse, however, healed the muscle injury tremendously as well. So very different than the old to old, and this was fascinating. So the researchers did a part 2. So in part 2, they wanted to see what were the cells actually doing the repair. Was it the stem cells? So they radio-labeled the stem cells in the young mouse. And then when they spliced together the circulation and did the same thing, they found that none of the radio-labeled substance actually showed up in the repair. So it was not the stem cells that were actually facilitating the repair. What was it? Eventually they deemed it to be from the exosomes that were circulating from the young to the old mouse, and this ended up being called the Fountain of Youth experiment. So as a result of studies like that, we have these Silicon Valley startups now. One of them is being funded by the billionaire Peter Thiel, where they take plasma from a young donor and give it to an older individual, hoping that some of those substances are going to provide an elixir of youth, for the older individual. So that could end up becoming a possibility eventually. All right, so how do exosomes get made? Well, as you saw, they are discharged from cells. And in this case, we want exosomes from stem cells because we want them to have all the information from the stem cell to then communicate in what they do, which I'll show you on the next slide. So here's a cross-section of an umbilical cord. All right, you can see there's two arteries and a vein, but all this gelatinous material around those is what's called Wharton's jelly. And that is where the very, very active mesenchymal stem cells are. Because exosomes are a product of stem cells, they're a byproduct of stem cells, we want to then culture the stem cells so we can get more and more and more exosomes. So these stem cells from the Wharton's jelly are cultured in a laboratory setting. This is an example. And here's a hood where you can do this in sterile conditions. And the exosomes themselves are then taken out and put into the exosome product. Now, here we are back to this same image. When you look at the exosomes as they get discharged from a cell, you can see inside them there are components. And these components consist of various proteins, micro and messenger RNA, as well as some DNA. And when you look at this a little bit closer, this is actually backwards. This is the endosomal vesicle that then accumulates the DNA, the RNA, various types of transcriptional proteins. Then they get discharged through exocytosis. And then the exosomes are floating around. They get attracted to, actually let's go through this. They get attracted to specific receptors. So it's not a random process how an exosome, as it's migrating through the bloodstream or saliva or what have you, how they get attracted to their target cell. These are receptors that indicate the cell is experiencing inflammation or some other need for repair. Okay, so the exosome gets invaginated. You might as well just call it getting eaten. And when it does that, it releases all of that information payload into the recipient cell. And what happens is it sparks off all these types of interactions. These are just a few of them that basically are cell-to-cell signaling. Okay, so because the exosomes have growth factors, cytokines, transcriptional proteins, micro messenger RNA, they spark off all these cellular responses, and it is truly incredible what they can do. Now, I have heard people describe using exosomes as cutting out the middleman of the stem cell, but that's actually a little bit of a misnomer, because if you look at it, what's actually happening is the stem cells are being used to create the exosome. So you're actually cutting out not the middleman, but the beginning man. And then exosomes are being used, directly for the recipient. So here's a small list of what's actually in exosomes. There's many, many more, but this will just give you an idea. There's growth factors, interleukins, cytokines, things like EPO, which helping with increasing blood circulation, platelet activation, new blood flow angiogenesis, new neurons. I mean, it really is incredible what these growth factors and all of these cell-to-cell communication components end up doing. So, as I mentioned, these come from Wharton's jelly, mesenchymal stem cells, and the umbilical cord is donated after a scheduled C-section. So it is there's a consent form, there's a hefty screening process for the mother, so it's a very safe and regulated procedure. And then those Wharton's jelly mesenchymal stem cells go to an FDA registered firm that then cultures them in a cGMP compliant fashion, very standardized and sterile, and the exosomes are the byproducts. So when you look at how many exosomes you're dealing with, here's a mistake that we see throughout the industry that we have not made, but this basically is a sizer graph. And what you see is that exosomes are very small. They're between 40 nanometers and let's say 140 nanometers. So a very finite size range. And the problem we see is that most labs will say, oh, we have, 1 to 200 billion exosomes per cc, okay? And the problem with that is, when you look at this range, there's a lot of other things that can be in that range. Not just exosomes. You can have cells that are dead because they lose their water and they get very, very small. You can also have these trash proteins that aren't actually doing anything, and they're not exosomes. So they give you these counts, and they're unrealistic. The way that we do it with the products that we sell is through markers. So there are 3 markers that attach to exosomes: CD9, 63, and 81. In this picture of immunofluorescence, those markers are seen blue, red, and green. Okay, so you can then deem how many exosomes are actually present per cc, and here they are broken out. Okay, so that's a very, very specific way of figuring out exactly how many exosomes there are per cc, much more accurate than otherwise. So here's just a sampling of studies that are been published on extracellular vesicles. In this case, exosomes are the type of extracellular vesicle we care about. So you can see here kidney, COPD, inflammatory diseases such as rheumatoid arthritis, diabetes, metabolic disease, more about rheumatoid arthritis, psoriasis and lupus, musculoskeletal diseases such as osteoarthritis, ischemic stroke, and then other neurodegenerative diseases have been looked at, MS, ALS, so on, post-stroke. None of these are actually approved by the FDA for usage. I just wanted to point out some of the studies ongoing and that have been completed. So when providers look at how do exosomes fit in, some people use them exclusively, others use them in conjunction with umbilical cord stem cells, and they look at that as a 1-2 punch. The exosomes give a quick benefit, it sustains for a while. Umbilical cord stem cells give a slower start and then a more sustained benefit. So that is the current thinking of a lot of providers. When it comes to aesthetics, many providers in med spas and aesthetics clinics use exosomes because they are so small. They don't care what needle size you have. 32 gauge doesn't matter. And the reason is that they're so small, they're 3 times smaller than stem cells, so they're not going to get lysed, as they go through a tiny needle. Key things to know: exosomes do not cause a rejection. They're very, very safe to use. They undergo the same quality assurance for safety as the stem cell biologics at the FDA Regulated labs. Once again, they're very small. Needle size doesn't matter, which is making them really, really exciting for aesthetics procedures. But we also see providers using them IV and into joints. They can be used by themselves. They can be used along with the stem cell biologic for the 1-2 punch, or they can be used with PRP. A lot of people do that for those aesthetics procedures. So here's our product that is exclusively distributed by Regen Suppliers. ReBellaXO is an exosome product derived from umbilical cord Wharton's jelly stem cells. There are 15 billion exosomes per cc, and the reason that's impressive is because they are characterized by surface markers, not just by the cell counter that you saw earlier. All right. Regen Suppliers is your one-stop shop for everything regenerative. Just visit us online at regensuppliers.com. ReBellaXO is the name of our exosome product, and also feel free to call us at 888-568-6909. We have all kinds of promotional specials going on at any one point in time, so you can often find that ReBellaXO does have a special associated with it. All right, thank you very much for watching.